My name's Edward Lestrange, and I'll be presenting on behalf of Agnes Deer, Ben Russo, Taylor Burton, Eve Muller, and myself. Our research topic is on the effect of phosphorus supply on Phaseolus vulgaris fabaceae seedlings. This research was motivated by an interest in the importance of phosphate in fertilizers for early seedling growth. The element phosphorus is one of the 17 nutrients that are vital for the growth, health, and yield of all plants, as well as having an important function in many biochemical pathways of plants. Phosphorus performs unique functions and is classed as a major plant essential nutrient, as it is required in relatively high concentrations. Phosphorus in the soil is acquired by plants through uptake by the root system. This is dependent on the form the phosphorus is in: soluble, slowly soluble, or insoluble. Insoluble phosphorus is not available to plants, and soils often contain inorganic phosphorus that is immobile and unavailable. Fertilizers are used as a way of supplying soluble phosphorus to plants efficiently to ensure they have adequate amounts for optimal growth. Deficiency in phosphorus can have major effects on plant physiology, including reduction in leaf number and surface area, and stunted shoot growth in early development. Studying the ecophysiology of the interactions between phosphorus and plants is extremely important, as it can have adverse effects on crop production. The aim of our research was to investigate the role that phosphorus has on the growth of common bead seedlings by using a nutrient fertilizer containing all required nutrients, and comparing this to a nutrient fertilizer lacking only phosphorus. We hypothesized that the removal of phosphorus from the fertilizer solution would cause a decrease in plant growth and leaf number, compared to the seedlings treated with the full nutrient solution, including phosphorus. Eight common bead seedlings were studied for 14 days under the same environmental conditions. They were screened to be of similar initial size, structure, and color. Subjects were split randomly into control and treatment groups of 4, and the initial measurements of seedlings were recorded. A complete nutrient fertilizer was used as the control, while the treatment solution was made using a phosphorus-free fertilizer. Both were diluted in a 1:10 ratio for application. All specimens were watered to help prevent desiccation on days 0, 3, 7, and 10. The seedlings were not overwatered to ensure nutrients were not lost from the system. 3 milliliters of the control and treatment solutions were applied initially and on day 7. On day 14, final seedling height and leaf area were measured, as well as chlorophyll fluorescence, which was the mean of 10 readings measured with a chlorophyll fluorescence analyzer. This graph shows seedling height of both the treated sample and the control samples at day 1, 7, and 14. There was no significant difference in final seedling height, with a p-value of 0.743. There was a statistically significant difference between final leaf area of the treated samples compared to the control samples, with a p-value of 0.003. Samples treated with the fertilizer without phosphorus had a greater leaf area compared to the control samples that had no phosphorus present. A p-value of 0.310 indicated there was no statistically significant difference between final chlorophyll fluorescence of seedlings treated with or without phosphorus. Our hypothesis was not proven correct based on our results. This suggests that the addition of phosphorus does not improve bean seedling growth. There were various limitations to our study, such as not keeping the seedlings hydrated, as it was found on day 3 that all seedlings were dried out. It is possible that the growth of our seedlings was affected by limited water availability. The validity of our conclusions would be improved by ensuring all necessary variables are controlled during replicate studies. Future research would benefit from incorporating various concentrations of phosphorus to determine an ideal range for optimal plant growth in bean seedlings. Determining how often phosphorus solutions should be applied and whether the results are reflected among the other crop plants would provide additional insight into the best use of phosphorus in fertilizer solutions. Here's our appendix, and thank you very much.